Today I am going to deliver a lecture on starch chemistry. So to understand it, first you have to know what is starch. Starch is basically a widely distributed storage form of carbohydrate in plants. It means when plants synthesize glucose in the by photosynthesis process, it is stored in plant in the form of starch. Now, starch is basically a polymer of alpha D glucose molecules. It is composed of two different polymers. One is amylose and another one is amylopectin. Now, you have to know regarding the properties of these amylose and amylopectin as well as their differences. Now, first to start with the amylose. Now, the amylose is the straight chain of glucose molecules which are linked by alpha 1,4 glycosidic bond. It means the first carbon of one glucose molecule and the fourth carbon of another glucose molecule are joined through this 1,4 linkage. Now, the number of glucose units, it varies between the varieties of grains, and the chain length of amylose molecule may vary from 250 to about 350 glucose molecules. And usually the long molecules cannot present in straight chain form. They are coiled into a alpha helix form. The amylose molecules have molecular weight approximately about in the range of 10 to the power 6, and almost all starches contain 25% of amylose. Now, this is a structural representation of amylose molecule. You can find here that individual glucose molecules are joined through a straight chain, and when a portion of this amylose molecule is shown through the glucose units, you can find here also that one or first carbon of one glucose molecule joined with fourth carbon of another glucose molecule to form this polymer. Now, next comes the amylopectin molecule. The amylopectin molecules are very large and they are highly branched molecules as well. The amylopectin molecule consists of glucose molecules which are joined through alpha 1,4 glycosidic bond, and the branching is due to alpha 1,6 glycosidic bond. Now, the branches are relatively short and contain about 30 glucose molecules, but the molecular weight of whole amylopectin show that 1,000 or more glucose molecules are combined together in a single amylopectin molecule. Now, the branches of amylopectin molecules are clustered and they occur as double helix. The molecular weight usually varies from 5 into 10 to the power 8, and you can find it is much higher than the amylose molecule, as amylopectin contains more number of glucose here. Now, this is again a pictorial representation of amylopectin molecule. So you can find individual glucose molecules are joined through alpha 1,4 glycosidic bond and branching through 1,6 glycosidic bond, and accordingly the structure is represented here. Now, the we all know that starch usually form blue color with iodine solution. So this blue color of starch, which is formed by reaction with iodine solution, is the function of exclusively the linear chain, that is the amylose molecule. And the crystallinity of starch is the function of branched chain amylopectin molecule. Part of these amylopectin molecules intertwining with the linear fraction and form the double helix. Now, the amylopectin is present in all starches, almost all starches, and it constitutes about 75% in most common starches. And some starches consists entirely of amylopectin, and they are known as waxy starch. An example of this type of starch is waxy corn. Among all starch, potato amylopectin is unique among all starch because it contains more number of phosphate ester groups, and these phosphate ester groups are attached to the hydroxyl groups of starch molecule, and as a result, due to presence of these phosphate ester groups, there is increase in hydrophilicity and the water combining or water swelling power of the starch. Now, starch is present in the form of starch granules. Now, these starch granules are formed in the amyloplasts by deposition of different layers around a central nucleus called hilum. Now, the formation of the starch or formation of the layers can be controlled by both endogenous factor as well as environmental factors like light, temperature, etc. Now, the starch granules are made up of both amylose and or amylopectin molecules, which are arranged in radial direction and alternate layers. So, the starch granules contain both crystalline as well as non-crystalline region in alternating layers. So, crystalline region is due to amylopectin molecule, and non-crystalline region is mainly due to amylose molecule. Okay. So you can find here, this is a pictorial representation of starch granule where you can find that amylose and amylopectin molecules are arranged around the nucleus in radial direction and in alternate layers. Now the clustered granules or clustered branches of amylopectin occur as packed double helix, which are packed together to give the crystallinity of starch. Amylose molecules occur among the amylopectin molecules, and some of the amylose molecule can diffuse from the starch granule in partially water swollen granules. Now, the starch granules differ in size and appearance. Potato starch consists of relatively large egg-shaped or oval-shaped granules with a diameter ranging from 15 to 100 micron and are generally less dense and easier to cook. On the other hand, cornstarch granules have mixed shapes. Some are almost spherical, some are angular and intended, and their size usually ranges from 2 to 30 micron. Potato wheat starch also contains a diversity of size and ranging from 2 to 35 micron. Now, it is a pictorial representation of different starch granules. From this picture, it is very clear that potato starch is larger in size and almost uniform oval in shape. However, wheat starch or corn starch, etc., they have mixed shape and quite smaller in size than the potato starch. Now, next I will discuss regarding one important aspect of starch which occur during processing of food, and that is known as gelatinization of starch. Okay, gelatinization is basically formation of gel. So how it is formed and what are its characteristics? Now I will discuss it. Undamaged starch granules are completely insoluble in water, but it can imbibe water irreversibly. It means that the starch granules can swell slightly when they are added into water, and they can return to the original size upon drying. When starch granules are heated in presence of water, they undergo an irreversible order disorder transition, which is known as gelatinization. The temperature at which gelatinization occurs is known as gelatinization temperature. Now, when the starch solution is heated, some changes occur within it. Now, what are the changes? First, the hydrogen bonds binding the starch granules together becomes weakened. So since the starch granule is made up of amylose and amylopectin molecule, so hydrogen bond is formed between this amylose molecule or amylopectin molecule, and this hydrogen bond hold the whole starch granule together. So as a starch solution is heated, these hydrogen bonds are broken. As a result, the starch becomes weaker. As it becomes weaker, water can easily penetrate into the starch molecules, and as a result, the granules swell until their peak thickness is reached. Thus, during gelatinization process, water is absorbed by individual starch granules and held there tightly and causes the molecules to swell significantly. And it has been noticed that the starch granules can absorb approximately 20 to 40 gram of water per gram of starch. As a result, the viscosity of the suspension rises steeply and the crystalline structure of the starch is lost. Now, gelatinization takes place over a certain temperature range, and it is approximately 50 to 70 degrees centigrade, and this temperature is called as gelatinization temperature. And this gelatinization temperature is characteristic for each type of starch. It means in each and individual starch will gelatinize at a particular temperature. Generally, the larger starch granules like potato starch swell at what temperature, means gelatinize at what temperature than those with smaller granules like corn starch. So potato starch will gelatinize at what temperature, while corn starch will gelatinize at higher temperature. Now, the gelatinization temperature of different starch are as follows. Like potato starch is 56 to 66 degrees centigrade. For corn starch, it is 62 to 72 degrees centigrade. For sorghum starch, it is 68.5 to 75 degrees centigrade. And for wheat starch, it is 52 to 63 degrees centigrade. Now, during gelatinization process, a part of amylose molecule diffuses out or leached out from the granule and goes into the solution. Finally, the granules will burst or collapse. Starch swells or gels are composites of swollen gelatinized granules and collapsed granules, which are embedded in a continuous amylose network. In this network, amylose acts as glue or that holds the gel together. Now, this is a pictorial representation where the gelatinization temperature can be, or gelatinization process can be explained through pictures. So you can find first there is raw starch granule which are heated in water. Now, as it is heated in water, in the next step, it swell or increase in size. Now, after it, as heating is continued, the amylose molecule leaches out or removes out from the granule into the solution. And eventually the starch, some of the starch granules become collapsed. And finally, the leached out amylose molecule form a three-dimensional network within which the raw starch granules, Wolven starch granule, then the collapsed starch granule as well as water is enclosed or embedded, and this completes the process of starch gelatinization. Now, after gelatinization, if the gelatinized starch is heated above gelatinization temperature in presence of excess amount of water, starch granules further swell and eventually the mixture becomes very viscous and translucent, and finally it becomes a paste. Gelatinization is important as gelatinized starch are more easily digestible. Now, there are some factors that affect the gelatinization process. So, first one is the sugar. Sugar usually decreases gelatinization as well as strength because sugar competes with water. So, it hampers the gelatinization process. Next, the important factor is the acid. Acid can hydrolyze the glycosidic bond. As a result, the starch molecules become weak and it results in decreased gel strength. Starring also cause breaking of some of the glycosidic bond, and as a result, it decreased gel formation and gel strength. Next important thing, type of starch, and it depends on the composition of starch, the water content, initial water content in the starch molecule, size of the granules, etc. And finally, the temperature. Already I had mentioned that gelatinization is carried out at a particular temperature, and it is the important factor in this process. Now, after gelatinization, next important topic related to starch is called as retrogradation. Now, what is it and how it happens? Now, after gelatinization, when the hot starch paste is cooled rapidly, it forms a firm, rigid, and viscoelastic gel. However, if it is cooled slowly and stored, then starch molecules become progressively less soluble in the paste. And in this case, the amylose molecules, which are already diffused out from the granule into the solution, they start to lose water and bind together, resulting in the formation of some precipitate. These precipitates are known as spherocrystal. And the phenomena of this crystal formation or recrystallization is known as retrogradation. Now, the retrogradation of cooked starch depends on size of the linear component of starch, that is both the amylose molecule as well as the outer branches or linear portions of amylopectin molecule. So, both can take part in the retrogradation process. Amylose molecule usually undergoes retrogradation at a much more faster rate than amylopectin molecule because the whole amylose molecule take part in the retrogradation process. On the other hand, only the linear part of amylopectin molecules retrograde. Now the molecules rearrange themselves during this cooling process by losing water into a more crystalline nature. So retrogradation is basically formation of crystals. Now this picture represents different forms of starch or different conversion of starch. Picture A represents the raw starch molecules where both the linear amylose molecule as well as the branched amylopectin molecules are within the starch granule. And picture B represents the gelatinization process. Here you can find that some of the amylose molecules are coming out from the granule into the solution, whereas amylopectin molecules are still inside the starch granule. And picture C represents the retrogradation process. Here you can find in the outside the starch granule, the amylose molecules combine together or join together to form crystals. So it represents the retrogradation process. Now the retrogradation process depends on several variables like molecular ratio of amylose to amylopectin, size and the structure of amylose molecule, temperature, starch concentration, etc. Now, due to this retrogradation, many quality defects in food products are noticed, like staling of bread, loss of viscosity, and precipitate formation in soup and sauce. There's loss of crispiness in cookies, etc. So, among these, the most important one is the staling of bread. So, next I will discuss what is staling of bread and how it occurs. Now, staling refers to the undesirable changes that take part between the time the bread is baked in the oven and it is consumed. The staling of bread is noted by increase in crumb hardness, which becomes hard and dry. The crust, which are usually soft in nature, but it loses its crispness and gradually becomes leathery. So, the outer layer of bread is known as crust. It should be crispy and it should be slightly dry and hard, but due to retrogradation, it gradually becomes soft and leathery. On the other hand, the crumb should be quite firm and it should be soft, but due to retrogradation, the crumb becomes hard and dry. And also, there is loss in the perception of product freshness and the loss of quality. Now, how it occurs? Now, staling begins as soon as baking is completed and the product begins to cool. The hard crumb first appears just under the crust, and with loss of moisture, it progresses through the whole loaf. And after 2 to 3 weeks, the entire loaf has become hard and dry. Staling is more rapid at refrigeration temperature than at room temperature. Whereas in frozen storage, staling of bread is completely prevented. So that's why it is always advisable to keep bread in room temperature rather than keeping it in refrigerator. Now, staling is due to the retrogradation of starch. What happens during breaking of bread? The starch granules are swell and they are already gelatinized. Now, during this process, the linear fraction of the amylose molecules, they diffuse out from the starch granules and in the oven, they are combined together to form the crumb structure, and also there are some changes in the physical configuration of the amylopectin molecule. Now, as the bread cools, the amylose molecules link together to give the bread its soft, elastic, and tender crumb structure. The branched chain amylopectin molecules are still remaining inside the granules during baking process. But upon storage, the linear fractions or linear sections, that is the outer branches of the amylopectin molecule, cross-linked together slowly, and this results in increase in crystallinity. As a result, the bread crumb becomes increasingly firmer with time, and with finally the whole bread becomes hardened. The retrogradation is irreversible on heating. It means if the hard bread or stale bread is heated, it it is coming to soft again, and it recurs again when the one bed cools again. So therefore, retrogradation is not a serious defect. It actually loses the perception of the product, but it is not harmful for consumption. So with this, I am ending today's lecture, and thank you.